Hi, I'm Chloe Boulay, and I am going to be talking to you today about membranes. This is chapter 7.1 to 7.3 in the textbook. So what are membranes? Well, a membrane is basically just a permeability barrier surrounding cells and organelles. The membrane consists of a lipid bilayer with associated proteins. So in the corner there you can see a picture, and that is part of a typical lipid bilayer that is the basis of membranes. So membranes also define boundaries of the cell and any internal compartments in the cell, such as the nucleus, mitochondria, and chloroplasts of eukaryotic cells. So we're going to move on to the functions of membranes, which is chapter 7.1, and we're going to focus on the 5 key functions of biological membranes. So one of the first major functions of membranes is that they act as permeability barriers, and they define the boundaries of the cell and their organelles so that the inside of the cell is separated physically to keep the desired fluids inside the cell and the undesirable fluids outside the cell. And the permeability layer is basically just the plasma or cell membrane, which is a membrane that surrounds the cell and regulates the passage of materials into and out of the cell. So I forgot to in the last slide, but here is the permeability layer, and it is protecting the cell from unwanted sodium ions. And the second key function of membranes is that they serve as specific sites for biochemical functions. They have specific functions because the molecules and structures responsible are embedded in or localized on membranes. So there's the second one right there. So the third and one of the most important key functions of membranes is that membranes retain transport proteins. And transport proteins are membrane proteins that recognize substances with specificity and assist their movement across the membrane. They regulate the movement of substances into and out of the cell. So they basically control what is allowed in the cell and what isn't allowed in the cell. So nutrients, ions, gases, water, etc. are taken into the cell into specific compartments of the cell, and in turn wastes are removed, and the transport proteins do all of this work. So there they are right there, and they allow the necessities to come in and the stuff that the cell does not need or does not want to be removed. The fourth key role that membranes play in the cell is that they contain protein molecules that act as receptors to detect extracellular signals. And these extracellular signals are usually received through information from their environment through electrical and chemical signals. So there they are right there, and they detect information that the rest of our body is sending to our cells. So the fifth and final key function of membranes is that membranes provide mechanisms for cell-to-cell contact, adhesion, as well as communication. So there it is, the cell-to-cell interaction right here, and they can communicate from one cell to another. Okay, now we're moving on to 7.2, which focuses on the models of the structure of membranes. So researching membrane structure actually took a long time. So scientists and researchers tried to understand the molecular structure of membranes for over a century, and no one actually saw a membrane until the 1940s or 50s when electron microscopy was applied to study. So there you can see the lipid bilayer and the hydrophobic tails and the hydrophilic heads, and how the water is not touching the hydrophobic tails there, because two phospholipids come together to make a lipid bilayer. So basically, electron microscopy what is a technique used for gathering high-resolution pictures for biological and non-biological specimens. So you can see here, this is the membrane under electron microscopy, so you can see it there, and then you can see that the cell wall is about 5 nanometers thick. So now we're going to move on to talk about the fluid mosaic model. And this model is now used to describe all biological membranes. And the model is described as two fluid phospholipid bilayers with proteins in and on them. So at first it was described as a thin layer of proteins on each side of the phospholipid bilayer, but they later realized that the protein was globular instead of in a thin layer. So after years of research, it led to the findings of the fluid mosaic model of membrane structure, which was found in the 1970s. So here you can see this is the timeline of the development of the fluid mosaic model over the century. So some people might be wondering, why did they decide to name it the fluid mosaic model? Well, they named it that because the lipids and proteins were able to easily move laterally with the membrane, so it flowed very well together laterally, and they added mosaic because of the presence of proteins within the membrane, so they described that as a mosaic. So we're now going to talk about Charles Ernest Overton. So he was studying in the 1890s, and he worked with cells from the root hairs of plants. His major discovery was that nonpolar lipid-soluble substances penetrate cells and polar water-soluble substances do not. He also proposed that cell coats are likely a combination of phospholipids as well as cholesterols. And then we have Irving Langmuir, who studied a decade after Overton in the early 1900s, and he experimented with dissolving phospholipids in benzene and layered samples of them onto water surfaces. So he discovered that phospholipid molecules formed a lipid film 1 molecule thick, which is also considered a monolayer. He also discovered that phospholipids are amphipathic molecules and they orient themselves on water. So the hydrophilic heads face water and the hydrophobic tails orient themselves away from the water. And amphipathic just means that one polar hydrophilic end and one nonpolar hydrophobic end are what make up the molecule. After Overton and Langmuir, the next major advance came in 1925 when two Dutch physiologists took lipids from a certain number of erythrocytes, and erythrocytes are red blood cells, and these two physiologists, Gorder and Grendel, used Langmuir's method of spreading lipids as a monolayer onto a water surface to publish their findings. So Everett Gorder and F. Grendel. So they discovered that the lipid film on the water was approximately twice the estimated surface area of the erythrocyte or red blood cell. So they found that the erythrocyte's plasma membrane was made up of 2 layers of lipids. And this became the basic assumption for each successful finding to come after them in the understanding of membrane structures. So as we've seen in previous diagrams, we have the hydrophobic tails and the hydrophilic heads, and then the water out here, liquids out here, so that the hydrophobic tails are not being exposed out here. And this is the phospholipid bilayer here, so it's a double layer. So moving on to Davison and Danieli. Their model suggested that proteins are present in membranes and they have lipid bilayers that are coated with the thin sheets of protein. And they founded this in 1935. Here is their model right here. You can see the lipid bilayer and the protein sheets here. And the lipid protein sandwich was their main finding and the first detailed representation of membrane organization. So we recognize the importance of proteins in membrane structure with this model here. So even though the Davison and Danieli model was very important to the development of the fluid mosaic model, they did encounter some difficulties around the 1960s. So once more data became available, it started to contradict some of their findings. So the electron microscopy findings found that membranes were 6 to 8 nanometers thick. Lipid bilayer was 4 to 5 nanometers thick, which only left 1 to 2 nanometers for protein. So it was most likely in globular states. So it wasn't as Davison and Danieli found, where it was thin sheets on either side of the phospholipid bilayer. So this did not account for different kinds of membranes. So they didn't take into account the different types. So after Davison and Danieli, there came Robertson, and he suggested that all cellular membranes must share a common basic structure. And he also suggested that the space between two cells contains the hydrophobic region of lipid molecules, as you can see in the diagram here. So cell 1, cell 2, and then the intercellular space right here. And then he also found that a membrane consists of a lipid bilayer coated with sheets of protein on both sides. However, that was later proven to be flawed by the electron microscopy findings. So after Robertson came Singer and Nicholson, and in 1972, they found that a membrane consists of a mosaic of proteins in a fluid lipid bilayer. And actually, the problems with the Davison-Danieli model sparked interest in the development of new ideas on membrane organization, which climaxed in 1972 when Singer and Nicholson found this fluid mosaic model. And this is now the model that dominates our view of membrane organization. And it has two key factors. The model envisions a membrane as a mosaic of proteins embedded in or attached to a fluid lipid bilayer. So different from earlier where they were viewed membrane proteins as thin sheets of protein on the membrane surface, they are now seen as globular entities of protein within the lipid bilayer. And the fluid nature of the membrane is a critical feature of the Singer-Nicholson model. So most of the lipid components are in fluid motion. Now we're going to talk about Unwin and Henderson's findings. So Nigel Unwin and Richard Henderson used electron microscopy to determine the 3D structure of Bacteria rhodopsin at low resolution and to reveal its orientation of the membrane. And in 1975, they found that Bacteria rhodopsin consists of a single polypeptide chain, which was folded back and forth across the lipid bilayer a total of 7 times. So this is one of those chains here. There's a few over there. And those are the polypeptide chains folded back and forth to create that chain. So looking into microdomains, we can see that since Singer and Nicholson proposed the fluid mosaic model, it revolutionized the way scientists think about membrane structure. It launched a new era and envisions the membrane as a fluid bilayer of lipids with a mosaic of associated proteins. So as you can see here, it's just a lot of repetitive pictures here, but they all show in different detail. So yeah, this is the Singer and Nicholson fluid mosaic model. So we're going to move on to 7.3, which is the membrane lipids, the fluid part of the mosaic. Membranes actually contain several major classes of lipids, and these include phospholipids, glycolipids, as well as sterols. Looking into phospholipids, they are the most abundant lipids found in cell membranes, and they are critical to their role in the membrane structure. So here is a phospholipid, how it would look with the chemical structure here. Next, we're going to look at glycolipids, and they are formed by adding carbohydrate groups to lipids. And some are glycerol-based, so they are called glycoglycerolipids. Kind of a mouthful there, and this is the chemical chain there. And the last major class of lipids in membranes is sterols. And besides phospholipids and glycolipids, the membranes of most eukaryotic cells contain significant amounts of sterols. Cholesterol is the main sterol in animal cell membranes, but it's not found in the membranes of most bacteria. It is also absent from the inner membranes of mitochondria and chloroplasts. However, some bacteria contain sterol-like molecules. These are called hapanoids, and they are rigid 5-ringed compounds that act like sterols by adding stability to the membrane structure. So this is the chemical chain of a sterol right here. So now we're going to look at fatty acids, which are actually essential to membrane structure and function because of their long hydrocarbon tails. And most fatty acids in membranes are between 12 and 20 carbon atoms long. So in this figure, it shows several fatty acids that are common in membrane lipids. So palmitate, stearate, oleate, and linoleate. A little bit hard to pronounce, but there are the carbon chains there. We are now going to look at lipid analysis and lipid component of membranes. They are difficult to isolate and study because of their hydrophobic nature, and we use TLC which is thin layer chromatography, and this is an important technique for isolating lipids, and it is used to separate different kinds of lipids based on their relative polarities. So it's just a small diagram there. Moving on to membrane asymmetry. So most lipids are distributed unequally between the two monolayers that constitute the lipid bilayer, and it is established during membrane biogenesis by the insertion of different proportions of the various lipids into each of the two monolayers. It includes differences in both the kinds of lipids present and the degree of unsaturation of the fatty acids in the phospholipid molecules. So once established, asymmetry tends to be maintained because of the movement of lipids from one monolayer to another. It requires the passage of hydrophilic head groups through the hydrophobic interior of the membrane. This is thermodynamically unfavorable, and actually what happens is a transverse diffusion, so a flip-flop, and it occurs most frequently in natural membranes than in artificial lipid bilayers. This is because some membranes, in the smooth endoplasmic reticulum in particular, have proteins called phospholipid translocators or flypases that catalyze flip-flop of membrane lipids from one monolayer to another. So I already kind of talked about flip-flop in the last slide. It happens occasionally, but it's very rare. And here is a diagram kind of showing that flip-flop there. Now we're going to talk about something pretty important. So the lipid bilayer. Membrane lipids form a fluid bilayer that allows lateral diffusion of membrane lipids and proteins. So membranes function properly, but only in the fluid state. And actually lipid molecules move relatively quickly because they are smaller than proteins. And the lipid bilayer has a phase transition, which means that at a certain temperature it melts and becomes a fluid. So to function properly, a membrane must be maintained in the fluid state, and membrane fluidity changes with temperature, as you could probably assume. So as temperature increases, so does membrane fluidity and vice versa. So if the temperature would decrease, then so would the membrane fluidity. So moving on to fatty acid composition, a membrane's fluidity depends on the types of lipids it contains. So two properties of a membrane's lipid makeup are important in determining fluid. And this is the length of the fatty acid side chain as well as the degree of unsaturation. So looking more into sterols and membrane fluidity, the presence of sterols affects membrane fluidity for eukaryotic cells, mainly cholesterol in animal cell membranes. And sterols decrease the permeability of a lipid bilayer to ions and small polar molecules. They also regulate membrane fluidity, so most organisms are able to regulate membrane fluidity primarily by changing the lipid composition of their membranes. Looking into lipid rafts here, so lipid rafts are localized regions of membrane lipids that sequester proteins involved in cell signaling. They can also be called lipid microdomains, and they are dynamic structures that change in composition as individual lipids and proteins move in and out of them. So they play a big role in the detection of and response to extracellular chemical signals. Lipid rafts are involved in the transport of nutrients and ions across cell membranes, and the binding of activated immune system cells to their microbial targets, and the transport of bacterial toxins into intestinal cells. So many receptor proteins involved in the detection of external chemical signals are localized to the outer lipid monolayer of the plasma membrane. So looking at this diagram here, these are lipid rafts, and this is showing them in a lipid bilayer setting. So now that we have covered 7.1 to 7.3, we are going to go over some questions that may have come up within these three chapters. So question 1 is, each of the following is a type of cell-cell junction except A, adhesive, B, tight, C, gap, D, plasmodesmata, and E, all of the cells junctions. And the correct answer is E, and I could not figure out how to get the arrow pointing in there at a different time, but yeah, E is the answer there. So all of these are cell-to-cell junctions. So the next question asks that each of the following is a function of membranes except, so A, defining cell and organelle boundaries, B, sites for specific biochemical functions, C, information storage, D, regulation of transport, and E, cell-to-cell communication. And the answer is C, information storage. So that is the only one that is not a function of membranes. So moving on to question 3, we're halfway there. So each of the following is a model for membrane structure except A, Overton lipid coat, B, Langmuir monolayer, C, Gorder and Grendel bilayer, D, Singer and Nicholson fluid mosaic, or E, Watson and Crick double helix. And the answer is actually E, Watson and Crick double helix is the only model that is not for membrane structure. So number 4, in response to temperature changes, cell membranes change state to become more solid or more fluid by undergoing A, lipid raft formation, B, membrane folding, C, a phase transition, D, transverse diffusion, or E, differential scanning calorimetry. And the answer is C, a phase transition, which is when the lipid bilayer is at a certain temperature and it melts and becomes a fluid. Moving on to number 5 here, which of the following molecules enters kidney cells via a specific transporter? A, water, B, carbon dioxide gas, C, cholesterol, D, transverse diffusion, or E, differential scanning calorimetry. And this one is A, water. It is the molecule that enters the kidneys via a specific transporter. So it is taken by specific transport proteins through the hydrophobic tails and through the phospholipid bilayer. And our last question, which of the following propose the sandwich model of membranes in which lipid bilayers are coated on both sides with thin sheets of proteins? Is it A. Overton, B. Langmuir, C. Gorder and Grendel, D. Davison and Danieli, or E. Robertson? And the first people to actually come up with this and the sandwich model was Davison and Danieli, when they thought that there was a thin layer of protein on each side of the phospholipid layers. And this was later proven to be wrong because there was not enough nanometer thickness in the cell membrane for there to be a thin strip on each side. So they found out later that it was globular protein entities. So I just wanted to thank you for listening to my presentation. I hope you enjoyed it, and yeah, I hope this helps everybody in Chapter 7.